The overall goal of this amputation procedure is to selectively and completely remove the pharynx from planarians without damaging other organs.
This method can help answer key questions in the regeneration field, such as how stem cells recognize missing organs and initiate their regeneration. The main advantage of this technique is that it is very precise and that other organs are not damaged during the process. I first had the idea for this method when I was anesthetizing planarians with sodium azide. I noticed that exposure to sodium azide caused the pharynx to pop out. Visual demonstration of this method is critical as the amputation requires the proper selection of animals as well as a careful observation of animal movement and morphology. Demonstrating the procedure will be Divya Shiroor, a graduate student from my laboratory.
Before beginning the procedure, use a plastic transfer pipette to select worms that were fed 5 to 7 days previously into a clean petri dish. Place a flat ruler under the petri dish to confirm that each planarian is at least 6 millimeters in length as they crawl across the dish. Then transfer up to 20 selected worms into a 35 millimeter petri dish and use a transfer pipette to remove all planarian water from the dish. Place the dish under a stereo microscope and adjust magnification such that multiple animals can be viewed. Add 5 milliliters of freshly prepared 100 millimolar sodium azide solution to the dish. Monitor the animals without moving the dish for 3 to 4 minutes to observe the extrusion of the pharynx. When at least half of the animals in the dish have fully extended a pharynx,